Welcome to NTP Outreach. The gross human brain is one of our most popular activities, and so we try to bring it to whatever outreach event that we're doing. And so the human brain, yes, this is a real human brain. You will get a lot of non-believers who are very adamant that this is not real. It is. So a couple of things that you get asked very often. No, we do not know what the gender is, whether this was a male or a female brain, and we do not know if this person, how old they were. We know that it came from a healthy individual who didn't have any sort of brain diseases, and we know that that individual donated their body to science so that people could learn more about the human brain. And so you'll get those questions a lot. And so the first thing that you'll want to do is ask the participant to put on gloves. Safety first. The brains are stored in just an alcohol water solution, but they have been fixed using formaldehyde, which is dangerous in and of itself. The brains aren't dangerous just sitting here, but you still want to have participants always be wearing gloves when they are touching the brain. And so you want to always use two hands for the whole brain, picking it up from either end. And so you'll want to sort of grade up or down your interaction with the participant based on how old they are. So if you are working with a child, you want to make sure that you're guiding them to how they should handle the brain. So you want to say, put your hands out like this, cupping them very close together, and then place the brain gently in their hands.
It's squishy.
It's squishy.
Ew.
So, Ryan, what do you think is the front and what do you think is the back?
I would say this is probably the front, and this is the back.
What makes you say that?
Because it seems like it would be oriented this way in someone's head. Because these look like they could be eye sockets.
Very good. So, when the brain is in your head, it's actually this way. So this is where your forehead is, and then these are where your eyes actually are.
Oh, wow.
And so this is your brainstem. This is what runs along your back and becomes your spinal cord. So is the brain lighter or heavier than you thought it would be?
It's a lot lighter than what I thought.
Yeah? Why do you think that is?
I would think that something that controls our every thought and action would be a lot heavier than, what is this, like 3 pounds?
Yeah. So the average human brain is about 3 pounds, which is really crazy when you think about how much blood the brain requires. So the brain at any one time is using about 20% of your body's total blood supply. And in fact, if we flip this brain over, you can even see one of the arteries. So your brain is made up of building blocks called cells. These specific kind of brain cells are called neurons. And neurons require oxygen and blood constantly. When they don't have oxygen for a significant period of time, that's what we consider a stroke or an aneurysm. And those cells will actually die. And so it's very important that our brain is using a lot of blood. So, something else that you can do that works well in a classroom setting is the 4 colors of the brain activity, which was created by NTP student Cece Lee. So, you ask the participants, the brain has 4 colors when it's inside of your head. What colors do you think the brain is? And you'll just have them yell out colors.
Gray.
Gray. Good. So, what makes you say that?
Because it looks gray right now.
It looks gray right now? So this brain has been fixed. That's why it looks sort of this grayish tan color. But when it's inside your head, there's actually a significant amount of brain matter that's gray. That's termed gray matter. So the cells that make up the brain, neurons, are composed of 2 different parts, the cell body and the axons and dendrites. And the cell bodies, when they gather together, actually look gray. And so we have gray matter in the brain. So what other colors do you think are in the brain?
Red.
Red, very good. So, like we talked about earlier, the brain is using about 20% of your body's total blood supply at any one time. A significant amount of blood is in your brain. And so that's why when most people do artistic renderings of the brain, it's usually pink, because the blood makes the gray matter and other colored matter appear pink or reddish. So what other colors do you think there are?
Is there blue?
No, there's not blue.
White.
White. Very good. So the other parts of the neuron, axons, transmit electrical signals called action potentials. And so if you've ever had an earbud where the wiring has gotten exposed, you know that electrical signals need insulation in order to travel from one place to another. Therefore, we insulate our axons with a fatty substance called myelin. And myelin is fat and is white. And so we have gray matter and white matter that we can kind of divide the brain into. Because much like cell bodies like to join together, axons like to join together as well. And so there's actually a fair amount of white matter tracts in the brain. And so these are where many axons of many neurons have bundled together in order to transmit electrical signals. And so the last color is usually the hardest for students to get. What do you think the last color of the brain is?
Well, there's gray and white, so I'm gonna guess black.
Black. Very good. So there's actually an area of the brain that is normally black, which is kind of crazy because we associate black with sort of dead things, and we would think that if the brain had a black area, it would be a bad thing. But these neurons are a very special population of neurons that naturally express the pigment melanin. And so melanin makes those cell bodies appear black in the brain. That area is called the substantia nigra. So you can ask the participants, have you ever heard of the disease Parkinson disease?
I have.
Yes. So maybe sometimes participants will know somebody who's had the disease or has had a relative had the disease. And so you can ask them if they know what happens during the disease. Most of them usually won't. They'll know maybe that there are motor symptoms associated with it. And so Parkinson disease is actually a disease where the body loses the dopamine cells. So these are also the cells that are expressing melanin. And so it's losing those black cells in the brain. So when you see a healthy brain next to a Parkinson disease brain, you'll see a big area of black sort of at the base of the brain, whereas in the Parkinson disease brain, you'll see nothing. You'll see cell bodies that don't express that pigment. And so this is an activity that works really well with elementary to high school students, and especially if you have an audience that you're having a difficult time getting them to sort of interact with you and get into the activity. This is a really great kind of kickoff to it. And so when it comes to the human brain, you'll be showing different audiences different things. So for the younger crowd, you can just sort of point out things like the wrinkles of this top. And so this wrinkly part on the outside here is called your cerebrum. So this is made up of cortex. And so the individual wrinkles are called sulci and gyri. And you can ask the participant if they know why the brain wrinkles up like this.
I don't know.
So usually if you have a comparison model next to you, you can grab that and point out that lower organisms don't have these wrinkles. So this may be a bird brain or a reptile brain, and it's usually very smooth on the outside. And so humans have such wrinkly brains because we are trying to make the most out of the space that we have in our head. So it wouldn't really make sense to have so much brain matter that is unfolded within the head, because we would have very large heads that would be very hard to carry around. And so our brain has actually evolved to sort of fold over itself. It's like crumpling a paper ball. You are making the most out of the space that you have by sort of decreasing the overall surface area. And in fact, the brain has folded itself in advantageous ways such that these folds will allow for better connectivity between particular regions. And so the human brain is extra wrinkly compared to other animal brains. At this point, since you have a comparison model next to you, you can ask them what structure looks the same amongst all of the different brains that you see. And so the answer is that the brainstem, so this region sticking out right here, is usually the same amongst all animals. And this is because the brainstem houses a lot of the important nuclei that we need for basic life processes. So this includes your breathing, your heart rate, digestion, all of these things that your body naturally does without you having to think about them are processed in your brainstem. And so no matter what animal you're looking at, it will have sort of a stick-like structure, and that's your brainstem. Another area that you can point out to even a younger audience is this extra wrinkly part here. This is called your cerebellum. And if you've had them do any of the other motor activities, in particular the prism goggles or the proprioception activity, or even the mirror box drawing activity, your cerebellum is what's responsible for allowing you to make those adjustments to your movement. And so the cerebellum, an interesting fact about it is that it actually has more cells in this portion than in the rest of the brain combined. And that's because these cells are extra tiny. They're called granule cells. So you think of like a granule of salt or of sand. We have granule cells in the cerebellum. And that's also what makes it extra wrinkly. So those are the main parts that you can point out to a younger person. And of course, something that works well no matter what the age is, if you have a participant that's still sort of lingering, but you've gone through showing them different parts, it means that they're still interested. And you want to keep them interacting with you by asking them about different things that they like to do. So Ryan, what are some things that you like to do?
I like watching movies.
You like watching movies? Yeah. So, you can point to an area of the brain that is involved in the activity that they like to do. In this case, you can talk about the visual system. So, if this brain were inside of your head, this would be the front, this would be the back. Your eyes are right here. However, you would think that if the brain wanted to make less work for itself, it would have visual processing areas right here. However, information from your eyes actually goes all the way back to here. This is a portion of your cortex called the occipital lobe. And the occipital lobe is actually where all of your information about your vision is traveling. You can even see here on the base of the brain, there's an X-like structure. So this is the optic chiasm. So your eyes have optic nerves that are sending information out, and it crosses here to sort of make a complement of what each eye is seeing in its own visual field. And so that optic chiasm continues to travel back to the back of the brain here, your occipital lobe. And so the occipital lobe has what's called ocular dominance columns. That means that there's areas that are stacked on top of each other, alternating which one is responding more to each eye. And so you'll have particular areas of the occipital lobe that are responding better to information from your left eye. And then you have other areas that are responding to your right eye. And so, what's another thing that you like to do, Ryan?
I really love listening to music.
Yeah, so you can point out the temporal lobe. So this is sort of the sideburn of the brain, and it has a few different structures. For listening to music, this is actually where the bulk of your auditory or hearing information is being processed. Another thing that is special about the temporal lobe, and in some cases you'll actually have this dissected out so that you can see it, but even if you don't, that's okay. The temporal lobe also has the hippocampus. Now this is something that was talked about in the mirror box drawing activity. The hippocampus is responsible for your declarative memories. So being able to experience things in the world and then process that into a memory that you can recall later is dependent upon the hippocampus. The hippocampus is also necessary for you to form spatial memory. So you can again talk about the HM case, a case of a man who had his hippocampus removed and he was unable to make new declarative memories. So he wasn't able to remember anything happening to him moving forward. And so the hippocampus is also responsible for spatial memory. So being able to remember, say, taking a right on a particular street to get to the coal center, things like that require an intact hippocampus. And in fact, there have been experiments done on cab drivers who are very experienced that show that they have much greater hippocampus activity than just a normal person because they use that area of their brain very often. And that gets into a slightly complex process called plasticity. So the brain is able to sort of rewire itself depending on the needs of the person. So, for example, if you're doing something that requires a lot of spatial navigation, you have to have a very well-functioning hippocampus. And if you are doing something where you have, a lot of visual adjustments that you need to make, you need to be able to compensate fairly quickly using your cerebellum. So if you're really good at sports, you must have a very complex and very developed cerebellum. And so plasticity can take form in particularly the way that neurons rewire itself to be able to talk to other neurons. A fun fact about the brain is that you actually have about all of the neurons that you're going to get by the time you're around 5 years old. Maturing and growing as a person actually depends on the way that those neurons are wired to talk to one another. And so it's the rewiring of neural networks that underlies plasticity and underlies being able to mature. And so a neuron has to be able to communicate in ways that are advantageous. So that means not too much communication and not too little. And so the brain actually undergoes a fair amount of pruning as people get older. So that means taking away connections that aren't really necessary and making sure that neurons are responsible for only the connections that they absolutely need to have. And so disorders of the brain actually are traced down to aberrant connectivity of neurons. One example is schizophrenia. So you see that schizophrenia patients actually have all kinds of neuronal connectivity that shouldn't be there. The same with autistic patients. They also have aberrant connectivity. And so the way that neurons are able to talk to one another is extremely important. So what's something else that you like to do, Ryan?
One more thing I like to do is work on math problems.
He likes to work on math problems.
Especially really hard ones.
Really hard math problems. So, when you're talking about the brain and you're talking about what makes us different from other organisms, is that we have a really complex thinking process. And so, you can point out, if you have a comparison model next to you, how the area that's most different between each animal is the cortex, this outer covering. So the fact that we are able to do things like solve complex math problems means that we need highly developed areas to be able to do that. And so the frontal lobe, so this part of the brain that's actually sitting above your eyes, is highly developed and allows you to make decision-making processes. And so particularly an area of the frontal lobe called the prefrontal cortex is really necessary in order to be able to focus our attention in on what it is that we want to do. And disorders of attention, so specifically attention deficit disorder or attention hyperactive deficit disorder, are due to the fact that the prefrontal cortex isn't well regulated, so it's not able to focus down your attention on things that you need to do. And so one area that gets overlooked sometimes is the parietal cortex. So the parietal cortex is what houses our cortices for motor and sensation. And they're actually right next to each other. So you see this very sort of well-defined sulcus here. That's the central sulcus. And so it divides our motor from our sensory cortices. And so your body is responsible for sending all of the information from nerves up to your sensory cortex. And likewise, your motor cortex has to send information down all the way to all of the nerves that are controlling your muscles and your joints and the things that allow you to move. And so the sensory and motor cortices have their fibers, so fibers are another word for axons, traveling through this portion right here called the pons. So the pons is sort of a large bulge that's coming out of the ventral surface of the brain. And eventually it funnels down all of those axons into the brainstem. And the brainstem is what becomes your spinal cord and runs down your back. So your spinal cord is both receiving information and sending information out. And that's why injuries of your spinal cord will generally result in paralysis, which is loss of sensation to a particular area and the loss of the ability to move that area. And so some other fun structures that you can point out on the brain. So here are the olfactory bulbs. The olfactory bulbs are responsible for information of smell. So information from your nose travels up to your olfactory bulbs and is processed and sent down to other areas that are controlling sort of our emotions and our feelings. Fun fact, the olfactory bulbs and your hippocampus, again this is in your temporal lobe, are the only two areas of the adult human brain that are known to undergo neurogenesis. So neurogenesis is the birth of new neurons. And you might remember from earlier that I mentioned that the brain doesn't make new neurons past a certain age. And this is true except for those two areas. This is also a fact that's currently being disputed by scientists who say that neurogenesis happens in other areas of the brain as well. And so things like that underscore the reason why we need so much research into the brain, because there's still so much that we don't know yet and that we need evidence in order to be able to know. So another fun thing that you can point out about the brain, other than naming the different lobes of the cortex, is being able to take the hemisection of the brain and point out specific sulci and gyri. And so one in particular that overlies the corpus callosum, which is another structure that you can point out, is the cingulate cortex. Now, this is a cortical area that's responsible for sort of our emotional processing and being able to attribute a particular feeling to a particular sensation. So, this is our corpus callosum. Now, the brain is made up of two equal halves. You can see it here in the whole brain, and those halves have to be able to communicate to one another. And they do so by sending axons. So again, these are the fibers that allow electrical signals to cross axons through the corpus callosum. So this is actually made up of millions of axons that are traveling to the other sides of the brain. And this allows the brain to be able to coordinate between its right and left halves. So sometimes people who have a disorder called epilepsy, which is a seizure disorder, have what's called a callosectomy. So that means that this corpus callosum is severed so that the seizures aren't allowed to spread to the other half of the brain. And these patients actually experience fairly normal lives. So they're still able to do complex motor processes and they don't have any cognitive deficits. However, they will oftentimes experience sort of a split brain dissociation, where one side of their body is doing one thing and the other side is doing another. So imagining getting ready for work in the morning and your right side of your body is putting on one pair of pants and your left side is putting on another. That's one of the examples of what a split brain patient looks like and what they experience. So something else that you can point out in the half section of the brain is particularly the structure of the cerebellum. So the cerebellum has what's called the arborization, and that's where this sort of becomes very tree-like. So the arborization of the cerebellum is basically allowing the very small cells to be able to be closely housed and do population responses. And so the half section is particularly useful for looking at different fiber tracts. So you can see that there are fibers that are directly going into the cerebellum, as well as fibers that are going out down through the brainstem. And so this is where you can kind of see the difference between gray and white matter, which is again difficult to do in a fixed brain, because you can see that there are definitely white matter and then slightly darker bundles of gray matter, and those are the brainstem nuclei that are responsible for our most basic processes. And so somebody might ask, well, why is there a blank space in the brain? And so you can explain to them about ventricles and cerebrospinal fluid. So the brain has actually open spaces where it circulates what's called cerebrospinal fluid, or CSF. This also is what's flowing over your spinal cord as well. So if anybody has ever had a spinal tap, that's what is being collected. CSF is responsible for a lot of different things. It's very important for ion regulation, keeping the neurons well fed, allowing the neurons a place to send their discreted contents. So CSF is important to flow through the brain, and these areas that it flows through are called ventricles. So your brain has 3 different ventricles. It has lateral ventricles, which you can see here, the very large lateral ventricle. You have here the third ventricle, which sort of looks very horn-like. And then you have the fourth ventricle, which is right over the brainstem. And so that actually exits out and forms a layer of CSF around the brain. So that gets into mechanisms by which we protect our brains. So we have very thick skulls, very thick bone outer covering of our brains. and that's to protect the head against trauma. However, when you experience trauma, you want to make sure that your brain isn't absorbing all of that shock. And so the cerebrospinal fluid helps to keep the brain buoyant so that when you hit your head, it's not absorbing all of the shock of that hit. So another thing that allows for the brain to be protected is the meninges. And so depending on your brain, you might actually be able to see one of the meningeal layers over top of it. So there's this sort of translucent white outer covering. This is your dura mater. So it looks a little bit flimsy now, but when it's inside of your head, dura mater is actually the toughest outermost layer that is underneath the skull. Dura mater is Latin for tough mother. And so underneath that, there are another layer called the arachnoid layer. And this layer actually has projections into it that are secreting cerebrospinal fluid. So this is where cerebrospinal fluid comes out of the fourth ventricle and joins underneath the arachnoid layer. And then under the arachnoid layer, there is the pia mater. And this is a very thin single-celled layer. And so your brain has a lot of different ways that it protects itself. The meninges being one, your skull being one, your CSF being one, and also your blood-brain barrier. So the blood-brain barrier allows us to keep our brain safe from infections that our body is experiencing because it basically filters out contents from the blood that shouldn't be allowed to pass through the brain. So if you have an audience that is particularly well versed, so if you're visiting a high school classroom, you can also talk about the limbic system. And so the limbic system is made of structures that are mostly within the middle of the brain. So your hippocampus is technically a part of the limbic system. Another structure deep within the temporal lobe and sort of able to see around here is your amygdala. So the amygdala is what we call the seat of our emotions. It's what's responsible for helping to attribute a fearful response to a particular stimulus. And so the amygdala is a target of many different disorders, particularly things like anxiety disorders and post-traumatic stress disorder primarily affect the amygdala. And so that is an area that scientists at the UW are currently targeting to see if they can modulate in order to help treat these disorders. Another area that you can kind of see here is your hypothalamus. And so the hypothalamus is a very important region for sort of our baser processes. So things like hunger, thirst, temperature regulation, sleep cycles are processed within our hypothalamus. And so this is a structure that is pretty similar amongst many different organisms because, again, it's tapping into those processes that are common amongst many living beings. And so those are sort of the main structures that are making up our limbic system. And so there are a lot more things that you can talk about from the brain that you'll be able to sort of pull from your audience. If you are visiting a school, you can also talk about your research and particularly do, sort of a special feature of brain regions that you study. You can also sort of draw upon from your audience things that they want to know about. So sometimes, somebody will have a burning question that they've always wondered how the brain does this. And so you may not always know the answer to those questions, but you need to be able to improvise and sort of say, well, this area does this, and it communicates with this area, so the thing that you're asking about is probably processed in region X. And the whole goal of outreach is to make somebody feel good about learning about the brain. And it's to make yourself feel more confident as an educator. And so if you don't always know the answer to a question, you can say, I don't know. Or you can sort of improvise and try to work your way to some sort of answer. But outreach is all about having fun, both you and your participants. And so if you don't know the answer to a question, that is perfectly fine. And so make sure that participants are always very gentle with the brains. Remember, these were donated by somebody who once had this in their body. And so we need to treat that donation with respect. And so you want to make sure that nobody drops the brain. So if they're holding it, keep their hands together at all times. Make sure that they're not pulling on things or sticking their fingers anywhere. So make sure that the brains are well taken care of. If you are doing something where you're having the brain out for a long time, just kind of, have some fluid in the tray so that you can kind of rotate it through so that the brain doesn't dry out. If you have periods where you can put it back into the bucket and allow it to sort of re-moisturize, please do so. But otherwise, just remember, it's all about having fun, and whether you're a freshman in college or a fourth-year graduate student, there are always things that people won't know about the brain. And that's one of the things that makes the brain still a great mystery to us, and one of the reasons why we have to continue to do research to learn about the brain.